Today, we'll be discussing translation in prokaryotes. The information stored in DNA is transcribed into mRNA during transcription and then is translated into proteins by translation process. So translation involves translating the sequence of mRNA molecule to a sequence of amino acids during protein synthesis. Ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after the transcription of DNA to RNA in the cell's nucleus. So this entire process is called gene expression, and translation is the final step of the gene expression. Translation occurs in three stages: enunciation, elongation, and termination. Tools of translation includes ribosomes, tRNA, mRNA, amino acids, various factors, and enzymes. Ribosomes are the cellular machine made of rRNAs and proteins that reads the mRNA and catalyzes peptide bond formation with small and large subunits. You can see here structure of a ribosome, smaller subunit, and this is larger subunit. The larger ribosomal subunit in prokaryotes contain three binding sites: A site, P site, and E site. A site means amino site tRNA binding site is a site where during elongation the incoming amino site tRNA binds. Then P site, the peptidyl tRNA binding site, is where tRNA linked to the growing polypeptide chain is bound. And the third one is E site, that is exit site. It's a binding site for tRNA following its role in translation and prior to its release from the ribosome. The tRNA is an adapter molecule that bring specific amino acid to the ribosome, matching mRNA codon with anticodon. Next is mRNA. mRNA acts as a carrier that carries the genetic information transcribed from DNA. It contains a sequence of three nucleotide units called codon, which specify the order of amino acids in the protein chain. Then amino acids are the building blocks of protein. Then various factors. Factors are actually proteins that help to start, to continue, and to stop the process. Three types of the factors are involved in translation process. They are enunciation factors, elongation factors, and termination factors. Enunciation factors in prokaryotes are of three types: IF1, IF2, and IF3. Elongation factors are elongation factor TU, elongation factor TS, and elongation factor G. Similarly, termination factors RF1, RF2, and RF3. Then enzymes. Two major enzymes involved in translation process in prokaryotes: aminoacyl tRNA synthetases and peptidyl transferase. Aminoacyl tRNA synthetases are here to charge each transfer RNA molecule by covalently linking it to its correct amino acid. Peptidyl transferase is responsible for catalyzing the formation of peptide bond between adjacent amino acids, thus building the growing polypeptide chain. Now we'll start with the translation process, and translation process completes in three phases: enunciation, elongation, and termination. Enunciation, when small ribosomal subunit binds to the mRNA molecule at the start codon. Start codon is AUG. The initiated tRNA means carrying methionine. So when it pairs with this start codon, this is start codon, and the larger ribosomal subunit join and forming a functional ribosome. Then elongation process. In elongation process, the ribosome moves along with mRNA reading codon, means three nucleotide sequence. For each codon, a specific tRNA brings the corresponding amino acids, and amino acids are linked together by peptide bonds, forming a growing polypeptide chain. You can see here, growing polypeptide chain. And in termination process, the process stops when the ribosome reaches the stop codon. So this is stop codon. Stop codons, we know three stop codons are there: UAA, UAG, and UGA. The release factor protein binds. This is release factor. It binds to the stop codon, and the polypeptide chain is released from the ribosome, and the ribosomal complex then disassembles. First or pre-preparatory step of the translation is amino acid activation. So amino acid activation involves three steps. First step is which is catalyzed by amino acyl tRNA synthetase, in which methionine is aligned to the tRNA methionine. The activation of amino acid involves the attachment of amino acid to the 3' end of the relevant tRNA molecule, and this process needs adenosine triphosphate, which is hydrolyzed into adenosine monophosphate and inorganic pyrophosphate. In the next step, this adenosine monophosphate AMP is released when the aminosyl adenylate intermediate is nucleophilically attacked, adding the aminosyl group to the tRNA at the 3-OH. The inorganic pyrophosphate is hydrolyzed by the enzyme pyrophosphatases to produce orthophosphate. And next, formation of aminosyl tRNA means following the activation of amino acid, the subsequent step involves the transfer of activated amino acids to the corresponding tRNA molecule. The transfer of activated amino acid to its corresponding particular tRNA is facilitated by amino acyl tRNA synthetase. The translation process consists of actually 3 stages, as I already told you. So this was the crucial stage, pre-preparatory stage, amino acid activation. Then the process of translation starts and consists of 3 stages: enunciation, elongation, and termination. So in enunciation process, that is the first step. In prokaryotes, the 30S ribosomal subunit attaches first to the enunciation factor IF3 in the means first step. So this factor prevents the 30S from rejoining 30S subunit from rejoining to 50S subunit. The 16S rRNA molecule also participate in this process of binding to mRNA. It accurately aligns the mRNA at the initiation codon, that is AUG, to initiate translation. The 30S subunit serve as a binding site for IF3, and the 16S rRNA is also involved in mRNA binding. Then next is recognition of mRNA. A small subunit of ribosome called 30S binds to the mRNA's Shine-Delgarno sequence, SD sequence. Now, what is this SD sequence? It's a short, preserved, purine-rich sequence. You can see here, this is mRNA and this is the Shine-Delgarno sequence of mRNA. And that starts a new basis. This Shine-Delgarno sequence starts a few bases before the AUG start codon. I can see here, this is start codon AUG, and this is Shine-Delgarno sequence. And this sequence binds with the complementary base pair UCC. I can see here on the 16S rRNA, UCC, located at the 3' end of a 16S rRNA. It anchors the 30S subunit of the ribosome to the correct position of mRNA. Then, during enunciation, a special initiator tRNA bearing N-formyl methionine is linked to the P site of the 30S ribosomal subunit with the help of enunciation factor IF2, which forms a complex with GTP. So, the IF3 is released as the formyl methionine tRNA mRNA pairs with the start codon AUG in the mRNA, and at this stage the 30S ribosomal subunit IF1, then IF2, GTP and F methionine tRNA mRNA formed, and this is the initiation complex. And finally, large subunit 50s binds to the 30s initiation complex, and this attachment is mediated by GTP hydrolysis, and all three initiation factors are released. Next step is elongation. Elongation completes in three stages. First is monocyl tRNA binding, second is formation of peptide bond, and then translocation. Monocyl tRNA binding means first this formal methionine tRNA binds to the P site, while other tRNA binds to A site and shift to P site during the initiation of prokaryotic translation. Then formation of peptide bond, the second step. Peptide bond formation occurs between the two amino acids that are located on the A and P sites of ribosome, and the peptidyl transferase enzyme catalyzes this peptide bond formation without the need of energy. Then translocation, and this is the last step of elongation cycle, in which ribosomes moves along the mRNA codon at the time forward mRNA 3' and this movement forward in the 5 to 3 direction is called translocation. Due to this ribosomal movement, the anticodon of dipeptidyl tRNA moves from the A site to the P site, and the decylated tRNA is transferred from the P to E site, and finally discharged tRNA is released from the cytoplasm for recycling. This elongation requires initiation complex, amino-acyl tRNA, elongation factors, and GTP. Next is termination. So termination process initiate when the ribosome meets any of the three stop codons. Stop codons are UAA, UAG, or UGA, and halt the peptide formation during translation process. So RF1 is responsible for recognizing the UAA and UAG codons, while RF2 identifies the UGA and UAA codons. The fully synthesized polypeptide is removed from the ribosomes, followed by the separation of the tRNA, mRNA, ribosomal subunits, and release factor.